Hey guys, so today I wanted to go through the standard level content of the topic of photosynthesis as part of the IB biology curriculum. So, the first thing with photosynthesis is learning the equation. This is the formula equation, I guess, and it would be 6 carbon dioxides plus 6 water molecules using light as the energy source essentially. Would turn to 1 glucose molecule and 6 oxygen molecules. But because oxygen is diatomic, you would have O2 instead of just O. If they ask you for a word equation, you don't have to specifically state that it's 6 water molecules, 6 carbon dioxide molecules. You just have to put carbon dioxide plus water turns to glucose plus oxygen. So yeah. So what organisms can photosynthesize? These organisms include plants, so trees, ferns, grass, flowers, things like that. But then you've got certain bacteria as well that can photosynthesize. And those are algae, just to name a few, and also which algae are found like in the sea, and they were the first things to ever photosynthesize. And you've also got certain types of bacteria called cyanobacteria, which if you also do higher level, you should know, or maybe you haven't studied this, but you would definitely know it from the endosymbiotic theory, because the cyanobacteria are theorized to then have evolved into chloroplasts. Standard level, don't need to know this, just an interesting fact. But another key word that I want to mention with photosynthesis is autotrophs. Can't spell. Autotrophs. And they're called autotrophs because they can essentially make their own energy and by making their own glucose. So they can use this glucose that they make during photosynthesis to then use as respiration. Auto meaning self, so autotrophs as in like they can produce their own organic molecules, which would be the glucose molecule in this case, through inorganic molecules. CO2, H2 are not organic molecules. So yeah, if you want to remember the word autotrophs, maybe you'll get an extra point for that. I don't know, just interesting. But when you're doing photosynthesis, there are two reactions essentially that we need to learn that happen, and those are light-dependent reactions and light-independent reactions. And the light-dependent reaction is essentially photolysis. Photolysis is the breakdown of the water molecule into its ions and oxygen. So essentially what happens is the light will break down the water molecule. That's how it happens. So what happens is you get H2O, and then you will get H plus, so a hydrogen ion, plus its electron. So that hydrogen loses its electron, and therefore you get the electron, and then just the water molecule. So I don't think for biology you need to balance the equation, but just in case they ask you for an equation for photolysis, because there's 2 oxygens here, you would need 2 oxygens here, and because you've got 4 hydrogens here, you need 4 here, and then also 4 electrons, because 4 of the hydrogens are losing 1 electron each. That's just a little bit of chemistry, but that is how photolysis works. And photolysis, what it additionally does, these light-dependent reactions, is they form 1 ATP. I don't know if we have to learn this for standard level, but it's just a little extra Googling that I did. And that ATP allows for the reaction to happen. So the reaction of bonding carbon and water, or that hydrogen ions now, to make glucose. and that would be the light-independent reaction. So the light-independent reaction would be the carbon dioxide with the hydrogen ions to then form the glucose molecule. And you're wondering like, oh, like where does the oxygen go? Like what happened here? Well, through photolysis, you get a byproduct of oxygen. So the plant doesn't really need the oxygen in that moment, like obviously for respiration it does, but it doesn't need it in that moment. It's a byproduct. What photosynthesis is essentially trying to achieve is the glucose. But yeah, those are the light dependent and the light independent reactions, simply meaning that in the first reaction you've got light needed to break down the water molecule into the hydrogen ions and the oxygen through photolysis, and then you've got the light independent reaction, which is reacting the carbon dioxide molecule with the hydrogen to then make glucose. But then there are certain factors that determine how fast the rate of photosynthesis will go. And the first three common ones that you probably learned from previous years are light intensity, concentration of CO2, and temperature. So as you can see, concentration of CO2 and light intensity are pretty similar, but I'll still go through them one by one. So light intensity, essentially what happens is that it increases like gradually, pretty linearly like you'd expect, because there is more light, so there will be higher, like there will be more light that can do the photolysis, which again is a process that is needed to complete photosynthesis. So obviously there will be a higher rate. However, at some point it will plateau because the photosystems, which is how the photolysis is done essentially in the chloroplast, will become saturated. Or I guess not saturated is the wrong word, I guess, but they will become full. Like there will be no space. There will be no space for photosynthesis to happen anymore. Now, for standard level, I'm pretty sure we don't need to know what a photosystem is, so you don't have to worry about that. But just know that just because you increase as you increase the light intensity, there will be an increase until a certain point, because there will just not be any more space for photosynthesis to be happening, essentially. So then you've got the CO2 concentration, which is similar to light intensity, and that is because as there is an increase in CO2, there will be more reactants. CO2 is what is needed for the independent reaction, where it reacts with the H plus ion. So if there's more reactants, there's obviously going to be a higher rate. However, at some point it will plateau, because think about photosynthesis is a reaction. That means that there's enzymes involved, and at some point all the enzymes that work with the CO2 will become saturated. So it doesn't matter that there's more CO2 if there's not enough enzymes to catalyze the reaction, then obviously it's going to plateau, because the reaction is just going to stay the same. There's going to be the same amount of enzymes that are going to be catalyzing the same amount of CO2, which means the rate of reaction will stay the same. Then you've got temperature. So at low temperatures, you've got low kinetic energy. That means that the molecules will be moving around a lot less, which means that there will be less collisions between the reactants essentially. And obviously, if the reactions aren't colliding, that means that there isn't going to be a lot of reactivity happening. There's not going to be a reaction. Well, there is, but it's just not going to be the rate isn't going to be very high. However, as the temperature increases, there will be a higher kinetic energy. And obviously, this will be the opposite effect as low kinetic energy. Because as you increase the kinetic energy, that means the particles will move around more, and that means that there will be more reactants colliding, and so the rate of photosynthesis increases. And at a certain point, you'll reach the optimum temperature. And this is the temperature at which the enzymes and the reactants will be essentially the most rate of photosynthesis will happen. If you increase it too much past the optimum temperature, you will have this very steep drop. And that is because the enzymes will become denatured. So enzymes will have very specific temperatures that will need to be kept for their shape to be kept. And if that temperature increases, so this can be like temperature or pH, if the temperature increases too much, that will cause them to lose their structure, and that's what's called denaturing, and that will cause them to lose their function, which means that it cannot catalyze the reaction, which means rate of photosynthesis also decreases. The final one is my favorite one because I just think it's fascinating, and that is light color. I'll write it down because I don't think there's anywhere. So, what we need to know for this is the absorption spectrum and the action spectrum. And the good thing about that is that they are pretty similar. So, when we see color, we see what colors are being reflected and what colors are being absorbed, we can kind of figure out. So when you see something that is green, like a leaf, that means that there is green light being reflected off of the leaf and into your eye, and almost every other color, like blue and red, specifically for leaves as we're about to see, or as you can see right here, that means that it will absorb blue and red light essentially. And so we can get this absorption and the action spectrum. And just so you know, we have to be able to know how to draw these. So the x-axis and the y-axis are really important. The x-axis is always going to be the same for both of them, and that's wavelength of light, and it goes from 400 to 700 nanometers. So also remember to put in the units. And what you get from 400 to 500 is like bluish tones, bluish, purplish. And this is the most absorption that you're gonna get from a leaf. So leaves, I mean, here you've got different pigments in the leaves that absorb different amount of colors, lights, I guess, but there is clearly a trend here where blue is absorbed the most. And then you've got another peak, which is like the orangey red. And what happens here is if you've got another big absorption, so red/orange absorbed a lot as well. Absorbed. However, green is reflected a lot. Green is reflected a lot compared to like blue and red hues. Also, I forgot to mention what you put in the x-axis, the y-axis, sorry, in the absorption spectrum is amount of light absorbed. Or I guess you could just put light absorbed. But yeah, and what is important for you to remember in this graph is that the blue will always be higher than the red. So your graph should always look like this. Something like this. It doesn't have to be exactly proportional, it doesn't have to be perfect, but if they ask you to draw it, that's what it should look like. These miniature bumps over here, they don't matter that much. It's just that you get the general shape right. Now the action spectrum, instead of having how much light is absorbed, it's telling you the rate of photosynthesis compared to the wavelength of the light. So the x-axis stays the same, but the y-axis changes. And just the same as the absorption spectrum, you don't have to memorize it specifically, like what the different bumps are, but just know that the first one is higher than the second one. So it will always be that the red is lower than the blue. Luckily for us, it's literally the same graph. It's just these ones, I guess, have steeper inclines and declines than the action spectrum, but it's the same graph. So that's pretty good. Now, if you think about it, the action spectrum is the rate of photosynthesis. If you really need to think about why the action spectrum looks like this. If the blue light is the most light that is being absorbed by a leaf, that would obviously mean that there would be a higher rate of photosynthesis because, as we said before, an increase in light, aka an increase in light absorption, would lead to a higher rate of photosynthesis. It needs light to be absorbed to do photolysis. So that's why the graphs are so similar, because the more light is being absorbed, the higher the rate of photosynthesis can be. It's lower for green because green is reflected. So in an experiment, if you changed, for example, the color of light, if you use blue light, you would have a higher rate of photosynthesis. If you use red light, it would be slightly lower than blue light, but it would still be pretty high. And if you use green light, there would be little to no rate of photosynthesis because light is less frequently absorbed, or is absorbed less than the other lights. So I think that's pretty cool. And the last thing about photosynthesis, I'm sorry if that was really long-winded, I didn't mean for it to be, but that's that session. So the last thing about photosynthesis is chromatography. In chromatography, super easy, essentially what we can use it, or scientists use it, to distinguish between the different pigments in a leaf or in a plant. And so as you can see here, you've got carotene, which is orange, xanthophyll, chlorophyll A, and chlorophyll B, and they move from the bottom up. So essentially, what happens in chromatography is you've got this solvent at the bottom, and this is usually like an alcohol. I think maybe ethanol, but I'll correct myself if that's wrong. And you will have this marking, usually with a pencil, with a dot of a leaf stain. Or for example, what we did in our class is spinach juice, used from spinach leaves. So we grinded down spinach leaves. And then what will happen is the solvent will move upward. So it'll trickle upward. And depending on how much the pigment dissolves in that solvent, it will move higher. So obviously carotene in this case dissolves way more in this solvent than chlorophyll B does. And xanthophyll dissolves in the solvent less than carotene does as well. And depending on how far it moves, you can find what is the RF value. So essentially what the RF value is the retardation factor. And this is essentially, you can use this to calculate how far the pigment can move with the solvent. It's essentially like a ratio. This can be calculated, so the RF, value can be calculated by dividing the distance that the pigment traveled by how far the solvent traveled. Now, you can see it in this diagram. Keratin traveled quite far, pretty close to the solvent, which means its RF value is higher than chlorophyll B, which traveled very little compared to the solvent. So, yeah, that's it for photosynthesis. I hope this was mildly useful to you. If you need help with anything else, or if I didn't explain something correctly, please tell me. I want to improve on this. And yeah, I will leave the whiteboard down below as always. I hope this was useful.